In this video, we are going to discuss is about nephron and its type. Before we let into the video, in last video we discussed a question. Which blood vessel has maximum urea and which blood vessel has minimum urea? So, the blood vessel hepatic vein, as urea is synthesized in the liver, it has maximum urea. Minimum urea we find in renal vein. As urea is filtered and it is mixed with the urine, the remaining urea, it has to be mixed with the blood alone, will be in the renal vein. So, maximum and minimum is the answer for previous video question. Now, let us learn about nephron. So, what is nephron, children? It is the structural and functional unit of kidney. So, possible question, the structural and functional unit of kidney is nephron. So, how many nephron do we have? We have 1 million nephron approximately in each of our kidney. And these are filtering unit also. Fine. So, now you can find this is the nephron. Define the Bowman capsule. A cup-like structure is called Bowman's capsule. And inside we can find a tuft of capillary which is called glomeruli. And this Bowman capsule along with glomeruli is also called Malpighian body. Again a possible question. What is called Malpighian body? Otherwise called renal capsule or corpuscles. Yes. So this is about the Bowman's capsule, and you can find two blood vessels. One is called afferent arteriole. The other one we call it as efferent arteriole. So if you see, the blood vessels are not in uniform diameter. Afferent is somewhat wider, efferent is somewhat narrow. And of course inside you find glomeruli, we discussed. Then it is followed by PCT. So this is PCT. Proximal means in the beginning. Convoluted means what? A wavy form. Tubule. You know it's tube. So proximal convoluted tubule we have right in front of the nephron. Then which is followed by a loop-like structure which is called Henle's loop or hairpin loop-like structure. So it is having a descending limb which is thinner and ascending limb somewhat thicker. Okay, and there is a transverse connecting limb, and there is also a distal convoluted tubule. I hope you understood, children. As it is in the distal part of the nephron, distal, convoluted again wavy form, tubule is tube. So DCT we have. Then we have collecting duct, which of course collect the urine from various nephron. So this is the structure of nephron. We are going to discuss in detail also. Now coming to its type, there are around two types of nephron. So let us see the types of nephron. You assume this is the cortex portion. If the nephron parts are restricted to the cortex itself. So you can see a Bowman capsule, PCT, Henle's loop, distal and collecting duct. Everything in the cortex, it is called cortical nephron. On the other hand, if we see the nephron where the Bowman capsule, PCT, and Henle's loop, you can see the difference. The Early's loop is even found in the medulla. So it is called juxtamedullary nephron. So we have cortical nephron and juxtamedullary nephron. So this cortical nephron 80 to 85% we have, and juxtamedullary nephron only 15% of nephron. Especially in birds and mammals we could find this nephron, where the Early's loop is very long and protrudes in the medullary region. This is one difference. So Early's loop, here it is short, and here it is Early's loop is long. Now third point, if you see, one more change we can find, vasa recta. Vasa recta is a capillary which is looping around the Early's loop, is absent in cortical nephron, whereas we can find the vasa recta. So the capillary which is coiled around, this vasa recta is very well present in medullary nephron. So I hope you understood what is the difference between cortical nephron and medullary nephron. If you remember, children, how many number of nephron do we have in each kidney? Can you kindly put your answers in the comment box? Thank you all.